So moving on to topic 12.3. This area in the textbook is pretty dense, so I'm going to try to cover just the most important pieces for you. As long as you understand the general concepts, you don't have to worry too much about the math. But this section deals with linked genes. So we now know that our chromosomes contain hundreds of thousands of genes, with the exception of the Y chromosome, which contains relatively few genes. The rest have a lot of genes on them. And there are certain genes on the same chromosome that tend to be inherited together, and we call these linked genes. It should be noted these are different than what we mean by sex-linked genes. Those are genes that are linked to the sex of the individual because of the chromosome they're carried on. But linked genes are on the same chromosome and they tend to be inherited together. So we'll get into that. So you remember Morgan, he was the fruit fly man, and he was doing his experiments with fruit flies, and he eventually ended up coming to some conclusions about how genes can be linked by observing the inheritance of two different traits. So in his case, he crossed two different types of flies that differed in traits of body color and wing size. He studied gray body flies with normal wings, and he also studied black-bodied flies with vestigial, or basically short wings. And he found that these two colors and wing sizes are usually inherited together in specific combinations as a result of the parental phenotypes. So this demonstrated that these genes did not assort independently like simple Mendelian traits, and eventually after many tests he was able to deduce that these genes were on the same chromosome. So this is what you can kind of imagine the chromosomes of those flies looking like. This would be our wild type mother, who is gray with normal wings, and then we have our, but she's a little bit dihybrid because you can see she's got the wild type here and then the recessive phenotype here. And then the male, we have a black fly with vestigial wings, and he has both the recessives. Most of the offspring ended up looking like one of these two options. So you can see we got a lot of similarities between the two parents. This is different than the traditional 9:3:3:1 ratios we would expect if these genes were inherited independently. So, and then we also saw non-parental phenotypes observed. So this led to Morgan thinking a bit about what's known as genetic recombination, which is the production of any offspring who have a combination of traits different from either parent. So opposite from what you would see here, because you see we would get still a lot of gray with normal wings and black with vestigial wings. So this is what we saw a lot of in the resulting experiment, and you can see our results differ somewhat from what we would expect if the genes were on different chromosomes. We would expect they assort independently, and we would see about an equal ratio of them. But instead, we tend to see a lot more of them on the same chromosome, and so because of that, we were able to we saw a lot more of the parental phenotypes, and because of that, we were able to deduce that those genes were on the same chromosome. So you can see that here a little bit more closely. These genetic findings relate to the chromosomal basis of recombination, so we can talk a little bit more about that. Mendel, we know, observed that there were combinations of traits in some offspring that were different from either parent. And just for some terminology's sake, if the offspring has a phenotype that matches one of the parents, we call this parental types. And those offsprings that are different than the parental phenotypes, we call recombinant types, because they now have recombined genes. If the two genes are on different chromosomes, we would expect to see about a 50% frequency of recombination between those two genes. So, for example, if we were to cross these green wrinkled homozygous recessive parents with a yellow round dihybrid parent, we would expect to see both some parental offspring, so we would get yellow round and green wrinkled, but also some recombinant offspring in that we have yellow wrinkled and green round, which neither of the parents were. Morgan discovered that even when two genes were on the same chromosome, sometimes you could observe that they would recombine. And so in doing this, he realized that there must be something that breaks the physical connection between genes that are on the same chromosome. And we later discovered that this is a result of crossing over, which happens between homologous chromosomes, as you know, in meiosis 1, and it kind of mixes up the DNA a little bit. So what happened with our parent flies here, if we understand crossing over, is we notice you can see it better with the mother fly because her genes crossed over during meiosis 1, and now we have these recombinant chromosomes. The male chromosomes, even if they did go through recombination, it wouldn't show because he had the same genotype on both genes. But the female, we can see the difference. And so that's how we end up getting offspring who have different traits than the parents do. And this shows you a little bit closer up version. So one of the reasons why this is beneficial is it has to do with evolution. So we know now that the way that we get traits that are eventually going to be more beneficial and help the survival of any particular species has to do with random changes in the gene frequency. So for example, we have recombinant chromosomes. They bring alleles together and create new combinations of genes in the gametes that are different. And then as you recall, we have random fertilization, so we have even more variety of combinations that can be produced. And then natural selection works by picking those that have the fittest traits to move on, survive, and reproduce. In trying to determine how genes were linked, we have one other scientist named Alfred Sturtevant, and he was one of Morgan's students who connected a genetic map, which was essentially a list of genetic loci or the genes places along a chromosome. And his prediction was that the further apart two genes were, the higher the probability that a crossover would occur between them, and then therefore they would be more likely to be recombined. Whereas if genes were really close to each other, they most likely wouldn't cross over because they would travel together. And so they would be more likely to stay together throughout. We call the genetic map of a chromosome a linkage map, and this is the part where you don't need to get too technical, but you might see distances between genes represented as map units. So one map unit represents a 1% recombination frequency. So you can determine the distance between multiple genes by knowing the recombination frequencies between one. So for example, with these flies, we know this gene and this gene have about a 9.5% recom frequency, and this gene have 9, so between this gene and this gene we get 17. But that's just a little bit of information about how they map genes. Genes that are very far apart on the same chromosome can have a recombination frequency near 50%, which is about what we see for genes on different chromosomes. So this usually indicates that they are not linked in any way. If it's a low recombination frequency, we might be able to tell that they're linked. And so with this information, Sturtevant was able to make linkage maps of fruit fly genes, and now scientists have used this to make genes of all sorts of chromosomes. So you can see these are all the different genes on one of the fly chromosomes. There's probably many more than this, but these are just some of the examples of what they've studied. And you can tell that those genes that are very close to each other tend to be linked and inherited together. So you've got your vestigial wings and your black body. We saw those were inherited together. Probably these two are as well, these two are as well. These ones we'd have to do a little math, but then these ones on the far end probably are not as closely linked and will be recombined more often. And that's where we'll stop for gene linkage.